Let's jump right into this explainer because we are looking at something absolutely mind-blowing today. We're diving into the cellular internet. We tend to think of ourselves as single, unified beings, but the reality? We are a walking, talking ecosystem of trillions of independent cells. And for us to survive without a central microscopic brain pulling the strings, those cells need a flawless, high-speed communication network. Today, we're going to decode exactly how that incredible network actually operates. We've got a solid roadmap to demystify this whole topic. We're going to hit 1, how cells communicate, 2, anatomy of a signal, 3, 3 ways to talk, 4, polar versus nonpolar signals, 5, nuclear family receptors, and finally, we'll wrap up with number 6, the secret study hack. Section 1, how cells communicate, the survival network. So at the most fundamental level, every single living organism out there, from a tiny unicellular bacterium to you and me, has to process information and respond to its environment just to stay alive.
Now think about this for a second. If a sudden temperature spike hits, how does a microscopic cell actually know it's time to build heat shock proteins? If it doesn't synthesize those protective proteins in time, the organism is a goner. That life-saving command has to be delivered and executed entirely at the cellular level.
And that brings us perfectly to our core concept. Cell signaling is, quite simply, the biological mechanism used to communicate commands and generate a specific response. It's how cell A, which senses a problem, tells cell B, which has the tools to fix it, exactly what to do. Maybe the response is triggering exocytosis to release a molecule, or maybe it's synthesizing a brand new protein. It's all about that cellular action and reaction.
Moving on to Section 2, anatomy of a signal, hardware and messages. It's kind of fascinating when you break down the bare minimum requirements for this system to work.
Because, well, you can't have a conversation without someone speaking and someone listening, right? Think of the signal molecule, the ligand, as a text message. This could be a gas like nitric oxide, an amino acid like aspartate, or a big protein. But that message is useless without a receptor. The receptor is basically the specific phone listening for that exact text. These are usually proteins or glycoproteins, and they can be hanging out on the plasma membrane, floating in the cytosol, or tucked way inside the nucleus. Without that receptor, the message just falls into the void. But here is where it gets really interesting, the binding strength, which is dictated by 2 constants, Kd and Ka. Let's focus on Kd, the dissociation constant. This specifically measures the exact concentration of a signal you need to occupy 50% of the receptors. So a lower Kd actually means you have incredible binding efficiency. You only need a tiny, tiny trace of that signal molecule to trigger a massive cellular response. They're a perfect match. All right, Section 3, 3 ways to talk, the network architecture. Let's zoom out a bit and look at the actual network topologies our bodies use to transmit these chemical texts when the cells aren't physically touching. Let's condense some seriously dense biology here. First step is autocrine signaling. This is basically setting an alarm for yourself. A great example is a T cell releasing interleukin-2 to trigger its own proliferation, or bacteria using quorum sensing. Sadly, cancer cells also hijack this self-signaling loop to fuel their own unchecked growth. Next, we have paracrine signaling. Think of this as whispering to a neighbor. It's highly local. This is your nervous system using acetylcholine at synapses, or blood vessels constricting from serotonin and dilating from histamine or NO. Finally, endocrine signaling. This is the global radio broadcast. A pituitary gland hormone drops into the bloodstream and travels huge distances, but only the competent cells, the ones with the exact right receptor, will actually tune into the broadcast. Oh, and before we move on, we absolutely have to mention direct contact signaling. This is where cells literally give each other a physical handshake. The signal isn't released into the wild, it's held out right on the membrane. You'll see this with the famous delta signal binding to the Notch receptor, or LAG2 to Lin12, and of course, the incredibly metal-sounding fast signal linking up with the death receptor. Section 4, polar versus nonpolar signals, the border crossing. So not all of these chemical texts can just waltz into any cell they want. There's a strict border patrol. The cell membrane is made of lipids, which creates a huge divide. If you're a polar hydrophilic molecule, like a peptide or an amino acid, even a tiny one like the 3-amino acid TRH, you love water, which means you cannot cross that fat-based lipid wall. You are stuck ringing the doorbell on the outside membrane receptor. But if you're a hydrophobic, nonpolar signal, like a cholesterol-derived steroid or a gas like carbon monoxide, you just ghost right through the wall and meet your receptor waiting for you inside the cytosol or the nucleus. But hey, biology absolutely loves to break its own rules. It throws us these wild curveballs.
You totally expect a nonpolar molecule to have an internal receptor, right? Well, look at prostaglandins, or major plant hormones, like brasinosteroid, auxin, and gibberellic acid. They are nonpolar, yet they surprisingly use plasma membrane receptors alongside their internal ones. It's a classic trick question, so definitely keep that in your back pocket.
Looking back at those polar signals, these amino acids brilliantly dictate the entire show at your synapses. They act as the gas pedal or the brakes. Excitatory ones, like aspartate and glutamate, pry open sodium channels, flood the cell with positive ions, and fire things up. On the flip side, inhibitory amino acids like glycine and GABA open up chloride channels, flooding the cell with negative ions and effectively slamming the brakes on the electrical signal. Section 5, Nuclear Family Receptors, the Inner Sanctum. Let's see how all this builds when we look at the receptors that live deep inside the cell, just waiting to get up close and personal with your DNA. These internal receptors are complex, microscopic Swiss Army knives. The AB domain at the top is a variable region, but the C domain, that is the magic key. It's called the zinc finger, and it uses exactly 4 cysteines wrapped around a single zinc ion to literally clamp onto the DNA. Then you have the D domain, which holds the nuclear localizing sequence, or NLS, which is the literal VIP pass you need to enter the nucleus. And finally, the E domain is where they pair up, or dimerize.
So, how does this work in practice? Let's look at type 1 cytosolic receptors. Imagine the receptor is stuck out in the cytosol, handcuffed by an inhibitor called HSP90. It's trapped. But when a nonpolar hormone, like a glucocorticoid, slips through the membrane and binds to it, the HSP90 handcuff drops off. The receptor is free. It pairs up with a twin to form a homodimer, flashes that NLS VIP pass to get into the nucleus, and recruits histone acetyltransferase, HAT, to unspool the DNA and kick off expression.
Now, type 2 nuclear receptors are totally different. They skip the line entirely. They're already waiting inside the nucleus, physically bound to the DNA. But they have a repressor called HDAC sitting on them, keeping everything turned off. So the signal molecule, let's say a thyroid hormone, has to travel all the way inside, boot that HDAC repressor off, and tag in the HAT enzyme so transcription can finally begin. All right, Section 6, the secret study hack, reverse engineering. We've all seen those terrifying spaghetti monster pathway diagrams in textbooks. Arrow going here, arrow going there. It's overwhelming. But I've got a game changer for you, straight from the source material. Here is the trick. Reverse engineer it. Do not try to memorize it from the outside in. Start at the finish line. Step 1, identify the specific target gene being expressed. Step 2, ask yourself, what is blocking this? Is there an inhibitor sitting on it? If there is, you know you need ubiquitin ligase to chew that inhibitor up. Step 3, trace the activator's origin backwards from the nucleus to the cytosol. And step 4, find the original trigger, the receptor and the ligand. If you literally write down numbers 1, 2, 3, 4 working backwards from the gene to the membrane, that chaotic web turns into a super logical checklist. Let's lock in this fundamental truth. No matter how wild or convoluted the chemical pathway gets, the ultimate endgame of all this cellular communication is always the creation of a protein or an enzyme. Whether it's autocrine or endocrine, a polar peptide hitting the wall, or a steroid ghosting through it, the entire point of this fast network is to tell the cell to build a specific tool to deal with whatever the universe is throwing at it. So, as we wrap up this explainer, I want you to ask yourself a question. Right now, in this exact second, trillions of your cells are firing off chemical texts just to keep you focused and breathing. What invisible messages are they exchanging? Every single heartbeat, every thought, is the result of millions of precise microscopic handshakes happening in the dark. It is absolutely wild when you think about it. Keep exploring, stay curious, and what do you think your cells are trying to tell you right now?